Hey class, this is a continuation of lecture 19, so we'll call this part 2 of 2 for the lecture 19 PowerPoint slides, and we'll call it lesson 25 since this is the 25th uh, segment of the course. Okay, so we talked about gal regulation in eukaryotes, and we're going to continue on in eukaryotes talking about hormone regulation. So this requires a little bit of knowledge on your part from steroid hormones. So hopefully you know already from your intro biology class that steroid hormones are made from cholesterol, and that means that they're lipids, and so they're hydrophobic, and that means they can pass through the bilayer without a transmembrane protein. Okay, so here's a cell, typical cell. Um, it has channels that allows things to go through, like water. So that's called an aquaporin, which literally means water hole. Um, and if you want to bring in glucose, you need insulin, which is a non-hormone or a non-steroid hormone. So it's a peptide hormone receptor that sends a signal to build a channel, kind of like permease, to bring in glucose. So that's what's going on here. Hormones don't, steroid hormones, lipid based steroid hormones, can pass through the phospholipid bilayer without a channel. So examples of these would be testosterone or estrogen or corticosteroids. Um, anything that's synthesized off cholesterol. So that's what we're talking about. So those are the steroid hormones, right? They can pass through the cell membrane, bind to something called a steroid hormone receptor, which we're going to show in a minute, and that activates it, brings it into the nucleus, and then that can bind directly to the DNA. Remember, proteins can have a DNA binding site and a gene regulation site, so it could have a transcription factor together. So these hormone receptors are actually proteins that have different sides to bind stuff. There are peptide hormones, like we just talked about here, which is insulin. It never enters the cell, it usually just gives a signal. So it binds at the surface of the cell, activates the transmembrane enzymes. So this goes across, kind of like ringing your doorbell, right? So somebody rings your doorbell, that usually elicits a response from your dogs or you to get up or and check the door. Um, and then that produces secondary messages. So that's going to signal something in the nucleus to turn on the DNA to express it in the RNA and then export that to make a protein. All right, so no big deal. All right, so like I said, steroids are synthesized from cholesterol. This is typical 4 rings cholesterol. And then it's the functional groups that are added that make it different. Like testosterone has a ketone group here. Um, Progesterone has this functional group. So ketone located here, C double bond to O is a ketone. We forgot that. This is a carbonyl group. This is a methyl group. Anyway, I'm not going to expect you to know all that stuff. These are plant hormones, so basically I just want to show you that they're lipids. Okay, so genes that are regulated. So let's draw our gene up here, and we know a gene has a promoter, a coding region, and this is from previous chapters, and a termination region. So we're going to focus on the promoter. We know that the promoter has like a ta-ta box, and so usually this has a hormone response element. That's a piece of DNA. Um, it's shown here. So these are sequences, DNA sequences, um, that are located in the promoter, right, in the promoter region. These can be promoter distal or promoter proximal, but they're usually not enhancers or silencers, although they could be. Um, and so that binds the hormone receptor, which is a protein. It's a trans element. This is a cis element. And then that protein is going to have a DNA binding domain, so that DNA is going to bind here. It's going to have a transcriptional activating domain, so that's going to help the RNA polymerase turn on the gene. Um, it's going to have a hormone binding, so a hormone binding region as well. Like, so it would bind estrogen or testosterone. Um, all right. So these hormone receptor elements, these DNA sequences, usually have a lot of copies. It can be in enhancers, like I said, or it could be in promoter distal. It's usually not in the promoter core regions. Um, so, like we said, steroid hormones can pass through the cell membrane. So when the steroid is absent, the activator isn't present to turn on the gene, so the gene is shut down. Um, the receptor itself is a protein that's found in the cytoplasm. So I'm just going to quickly draw this because I know that I have another like professionally done drawing from a book in one of these slides. But so here we have our receptor. It's a protein. It has multiple regions. I'm just going to make it a linear thing, but it doesn't have to be linear. Here's the nucleus. And let's just say here, I'm going to blow this up. So this is the hormone response element. Let's just say it's estrogen. So I'll replace the hormone response element with an estrogen response element. And this will be an estrogen receptor. So estrogen is produced, or you're exposed to it, passes through the membrane and binds to the estrogen receptor. So this is a protein. Remember, this is a lipid. So it's going to bind here. Let's say that this is the hormone binding region. So estrogen binds here. Let's just make it a circle. Once that's bound, usually this displaces something we call a heat shock protein that sort of guards this in a competitive manner. So the heat shock protein um, is dislodged. Once that occurs, it changes the shape of this, sends a signal to bring it into the nucleus. So this whole thing goes into the nucleus. So this area is the DNA binding region. So that's the DNA binding region. We'll just make it a 3 helices and 2 loops. So this is the DNA binding domain, and then this is the hormone binding domain. So it's going to bring in the estrogen with it. This recognizes the DNA sequence for the estrogen response element, and then on this end, so this is DNA, and then this end is the transcription factor or transcription activating factor. We'll just call it TF. This is going to interact with the RNA polymerase to turn on the gene. All right. So anyway, that occurs in the nucleus. And this is just sort of a written explanation of that. So when the steroid is present, it displaces the chaperone protein. This is called the heat shock protein. Same name. It's just like, we have multiple names for money, like cash, uh, bones, dough, whatever, bucks. All right. So that displaces the chaperone, the heat shock protein. The steroid then binds to the receptor. The receptor itself then enters the nucleus, binds to the hormone response element sequence. So this is the cis. And then transcription begins because it has a transcriptional activating region. So that's kind of how it works, and I 100% know I know how to image of this. Okay, so this is just a breakdown of what I drew. Again, this is a cholesterol, so this is cortisol. It's a steroid hormone. Um, it has a receptor, right, that binds to the. It has a hormone binding domain, so this is going to bind here. This DNA binding domain is going to go into the nucleus and bind to the cortisol response element, cortisol response. And then this might be the transcriptional activating transcription factor region of it. So remember, these are lipids, steroids, so they can go through the cell membrane. Once in the cell, the hormone reacts with the receptor, which is in the cytoplasm, and then it gets transferred to the nucleus because it has to. It can't bind to DNA because in all eukaryotes, the DNA is found in the nucleus. So I've seen books that show it differently, that showing the uh, the steroid hormone receptor uh, that is found in the nucleus, like it's always in the nucleus, and so I assume that there's some sort of discrepancy if it's going through the phospholipid bilayer and the nucleus, um, or if it's just going through the phospholipid bilayer. And I've seen it both ways, so I'm not 100% sure which is true, but it doesn't really matter. Um, so this is basically just showing you. This is this is heat shock protein 90. Remember, this is a chaperone protein. It's called a chaperone because it helps proteins fold into place. And these are the same. They have the heat shock protein and the chaperone is the same thing. These are found in the cytoplasm, although some can be found in the nucleus already. So in this case, we have the activating domain, the DNA binding domain, and the ligand binding domain, which in this case is uh, glucocorticoid. So the heat shock protein 90 is bound to this ligand binding domain, which would normally bind to the glucocorticoid. Once glucocorticoids are present, it would displace the heat shock protein, which is shown here, and that hormone would then bind to this ligand. So, in this case, the steroid hormone glucocorticoids, once it's bound to that, it gets transferred into the nucleus. It's showing you this is that response element. So, this would be the hormone response element, like it would be called an estrogen response element if it responded to estrogen, or a testosterone response element if it responded to testosterone, and so on. And then this is the activating domain that's going to interact with the polymerase to turn the gene on. All right. So let's see, what else do I have in this slide? Okay, translocates the nucleus, is an activator, correct. DNA binding domain. Okay, so this is a zinc finger. We talked about those. It binds to the glucocorticoid response element. So this would be called the GRE, whereas estrogen response element would be the ERE, and testosterone would be the TRE, and so on. Um, and then that activating region acts like a transcription factor to stimulate the transcription of genes. Um, I don't care that you know this. The sequence doesn't really matter. Just know that it's called a response element for the exam. Okay, so this is another image showing you that what it would be like if it actually was already in the nucleus. It's the same thing. Um, so this is an alternative model. We have the heat shock protein here. Glucocorticoid comes in. It's already in the nucleus, so it has to pass through this membrane and the nuclear membrane. It dislodges the heat shock protein 90, binds to this. So this is the ligand binding domain and the receptor. Remember, that's a protein. Uh, this is the DNA binding domain of this protein. And then this is the, uh, well, this would be the transcriptional activating domain of this ligand. Basically, it's just the same thing that we talked about, except it may already be in the nucleus. Polypeptide hormones, they never enter. So, example of this would be insulin. So, polypeptide means they're made out of proteins. They can't enter into the cell because these are particularly ones are um, water soluble, so they can't go through the transmembrane domain, the where the phospholipid tails are, which you can't see with that drawing. Anyway, so this would be an example of insulin. It would bind, maybe bring these two receptors together, activate some phosphorylating mechanism. So this becomes phosphorylated, changes its shape, and sends a series of response factors, so transcription factors, nuclear transcription factors, to turn on a gene. In this case, it would probably turn on a gene to make a, well, it would turn on gene to make um, a transmembrane protein. So it would open up a channel in the cell to bring in glucose. And then this is where glycolysis would start. And hopefully you learn glycolysis in your intro bio class. Okay, so this is the only way that eukaryotes can regulate their gene expression. Remember that eukaryotes have histones. We talked about these already. We have the the histone, the basic unit of chromosome organization is the histone subunits. So H2A, H2B, H3, H4, which we've already covered, and then the linker DNA. So this is what we call the nucleosome, and then the histone itself is an octamer made up of 8 subunits, which we already covered. So we know that chromatin can be modified. We've talked about heterochromatin and euchromatin and all this other stuff. So basically, it's, to simplify it, it's like kind of closing and opening a scroll, which is the example I've already used. We've talked about the 30 nanometer fiber packaged nucleosomes already when we talked about meiosis and mitosis. So as the DNA relaxes, right, then the regions can be read. So in this phase, this is more like you would see in a chromosome that's undergoing mitosis, and so it's going to get many times shorter and thicker, and that's how you can see it under the microscope. So this would be like during mitosis phase, not interphase. In interphase, it's more relaxed, and you have areas between these nucleosomes where the DNA can actually be accessed by the proteins and read. When they're wrapped around the proteins, it's like a closed scroll and it's not easily accessed or read. So as we get to a more relaxed state, the DNA can be accessed by those proteins. So this is the minor groove, this is the major groove. So we're generally in the major groove. All right. Okay, so we can turn on and off genes by remodeling the chromatin. So we can condense the chromatin to turn off a gene and relax the chromatin to turn on a gene. Remember, these transcription factors are going to bind to a cis regulatory sequence. Remember, so cis elements are DNA sequences, and then this would be a trans element. Um, okay, so hopefully this is a review, but there are different kinds of bonds. We have ionic and covalent bonds, and covalent bonds are very strong. If we know these are shared electrons. So we can covalently modify histones on their tail. So histone, the proteins, have like a tail region. Um, and this is where modifications occur. So we can acetylate these things on the tail or methylate them. There's lots of things that we can do. And so there is a enzyme called HAT that stands for histone acetyltransferase. And when we acetylate this, it decondenses the chromatin. There's also a complex that can physically do this called the switch-sniff complex. Either way, we can open the scroll, right? Repress or decondense the chromatin, and then that allows the gene to be transcribed. Remember, we talked about the pre-initiation complex, which is all those transcription factors and the RNA polymerase coming together to turn the gene on, to read it into RNA. Okay, so this is how it works. The nucleosome, right? Remember, this is the octamer with the DNA on it. This is the linker DNA. So DNA is negatively charged, and hopefully you know this. This is another review thing. I think we've actually covered this as well. In fact, I know we have. So remember, the phosphate group has a negative charge on it. And so this is the number 5. This is the number 1, 2, 3. Here's the carbon, that's 5. This is 4 here. So DNA is negatively charged, right? And so that means that it's going to be attracted to the positively charged histones. And we've kind of talked about this. So that's how DNA is wrapped around. So DNA is negatively charged, histones are positively charged. And the reason it's positively charged is there's lysine residues. I don't care that you know where it's at, but let's just say there's lots of lysines, and lysine is a positive charged amino acid, right? So this is what causes histones to be positively charged. And you should know that negative charged DNA is going to be attracted to positively charged histones. So this is the normal way that things work out. And this slide is just showing you that we can acetylate or methylate these regions of these tails on these histones. In this case, this is H4. All right. So here's some modifications of histones. So we can acetylate and deacetylate histones. So here's the lysine residues on the histones. It's positively charged. Um, so if we acetylate this, then that positive charge goes away. So when they're deacetylated, they're positively charged. When they're acetylated, they're neutral. And so DNA that's negatively charged is going to be more attracted to positively charged histones than neutrally charged histones. So that is going to relax the nucleosome and allow that to um, be open so that DNA can be read. Sorry, I'm getting a call, so I'm kind of distracted here. All right, so we can deacetylate these. So this is a histone deacetylase. This is a histone acetyltransferase, which we just talked about. And so we can reverse this um, to make them positively charged again. So by doing this, um, by acetylating them, this is all part of this. That's kind of what this here is. So it reduces the affinity to DNA. DNA is not going to be as attracted to neutral charges. And that opens the chromatin, opens the scroll so you can read it. Um, it also helps binding sites, expose binding sites for other proteins like transcription factors or things that we're going to bind to like estrogen response elements or other cis elements, DNA elements that are required for transcriptional activation. Okay, so just remember histone acetyltransferase acetylates histones, and when they're acetylated, they're neutral, right? When they're neutral, they unroll and allow for activation of transcription, right? It's like unrolling a scroll. Uh, histone deacetylase, HD or HDAT, same thing. That deacetylates it, so it makes it positively charged, which makes the DNA have a higher affinity for it. And so it closes the scroll so you can't read it. And by doing that, it's going to reduce transcription or deactivate transcription. All right, so we can physically do this too through switch SNF um, complex. So it can be recruited by transcription factors like Gal4 to expose DNA to make the nucleosomes actually physically slide. So let's say that this is our promoter. It's hidden here. Um, but if we move, physically move the nucleosome over, now the promoter is exposed and the RNA polymerase can bind to it, and the RNA polymerase can push these out of the way. But it's never going to bind unless the promoter uh, regions for the transcription factors are that transcription factors can find and bind to those DNA sequences, those cis elements. Okay, so we talked about enhanceosomes already, and you guys know that this is a complex of different transcription factors um, and activators and silencers that assemble in the promoter region to help recruit the RNA polymerase. So in this case, there's a thing called GCN5. I don't care that you know that. Um, it's going to bind. Well, maybe I do. Maybe you should know it. So GCN5 complex, like it's a complex of proteins, recognizes and bind to the enhancer regions. This in turn is going to add acetyl groups to the nucleosomes in between. So drawing is showing you that it's adding acetyl groups. We know that acetylating this makes these neutral, and so they have a lower affinity for the DNA. So they're going to relax a little bit. Uh, Once that's moved, switch SNF and uh, a co-activator called CBP bind. So here's the CBP, here's the switch sniff, and these are physically kind of pushing these nucleosomes apart to allow the RNA polymerase to bind to the promoter region. And then once it does that, so it's nudging aside the nucleosomes, and uh, now the Tata binding protein can bind to the newly exposed Tata box, and that allows transcription to begin. So this is showing the Tata binding protein binding to the Tata box, um, and allowing the polymerase to start transcribing this region. Okay, so you can long term mark these chromatin regions for activation or inactivation. There's kind of a code, right? So there's 150 different modifications that can be done, more than 150 modifications that can be done to histones. And so you can imagine you have this histone tail, and we're going to have lots of different stuff, kind of like just kind of like the enhanceosome. So the enhanceosome is made up of complex parts. Again, we can have all kinds of different modifications to histones, and those are going to cause different effects. So these sort of codes, like these patterns, these histone patterns, these variable patterns, create regions of either active or inactive gene expression. So uh, we talked about heterochromatin or euchromatin already. So that should be a review. And if you don't remember, you can go back at previous lectures and look at it. So in the end, basically, what I'm trying to point out is that you can inherit DNA, or it can get modified through diet or whatever, smoking, lots of environmental factors. So you can inherit a perfectly good piece of DNA. Let's say that it makes your hair brown, right? But if it's acetylated or methylated in a certain way, then it's going to cause the nucleosomes to wind this around, and so that's going to shut off this gene. So even though it would normally make brown pigment and you would have brown hair, if it's turned off, you're going to go to the default, which would be blonde. And this happens a lot during pregnancy or other hormone responses, where perfectly good genes can get shut down, or genes that are normally shut down can get turned on. And it has to do with the way that these histones have these modifications on them. So hopefully that makes sense. um, you inherit this form of chromatin during DNA replication, so the expression behaves identically in daughter cells as it did from the parental cells. DNA methylation, so there's cytosine residues that are methylated. We call those CpG islands. The degree of methylation in cis-regulatory sequences is inversely correlated to gene expression. So, in other words, as we increase methylation, it decreases gene activation. So this is different. In acetylation, it turns the genes on, right? It relaxes them. In methylation, it turns the genes off. And so as we do more methylation, we get less activation of expression. Again, can be inherited, and it can during when so during replication phase, we have G1, S, G2, and M phase. During the replication phase, you can inherit the chromatin at replication, and it can be specific for specific tissues. Um, the enzymes that do this methylation are called methyl transferases. So we have uh, acetyltransferases. Those are acetylating. We have methyl transferases. Those are methylating. These codes, this series of methylation regions and histone acetylation regions and other modifications that we're not. That's beyond the scope of this class. If you want to know more, you can take a molecular genetics class. But we collectively call these epigenetic marks, right? Or epigenetic inheritance. So you can pass on a perfectly good gene that's shut down or being expressed from your parent, and this can be tissue specific and it can even be sex specific. So we're going to talk about that now. That process of inheriting this stuff is called genomic imprinting. What slide are we on? 54? Okay. Um, so imprinting is a term that you get, you inherit it from a parent. And it's not the DNA itself, it's the uh, pattern of chromatin acetylation, methylation, and other modifications. So the expression of the genes, right, are predetermined by the parental origin. So you could have paternal imprinting or maternal imprinting. Um, this can be done through sperm, right? So that would be paternal, or egg, which would be maternal. It doesn't matter what the DNA sequence is. So just like I said, if you have a sequence that codes for a brown pigment and it's turned off, you're not going to make a brown pigment. You will go to the default, which is blonde. Um, so this is well studied in mice, and there's a gene called IGF2. You need to know this. Um, so IGF2 stands for insulin-like growth factor 2. Um, in mice, only the copy that they inherit from the father is expressed. The one that they inherit from the mother is inactive. So if we say it's maternally imprinted, that means that in the mother it is shut down. And if we say it's paternally imprinted in the father, it's shut down. The maternal copy is inactive. That's maternal imprint. The paternal copy is inactive. That's paternal imprint. So there's another gene called H19. Only mom's copy of that is expressed. So it's paternally imprinted. That means dad's copy is not active. So the DNA in these regulatory regions of imprinted genes is methylated in a sex-specific manner when the gametes are formed. All right, this slide is just showing you that there are certain cells that produce gametes, and then in the gametes, there's certain cells that become sex cells. So this is where it occurs. It's only in gametes, right? It's not in all the other cells. All right, so this is how genomic imprinting works. Okay, so this is a super busy slide, but we're going to talk about the silenced, right? It's methylated. Remember, adding methyl groups turns off genes. Adding acetyl groups turns them on. So the black here is going to be turned off. The yellow is going to be turned on. The color codes are going to be from dad, right? So this is what this male inherited from his father. This is what this male inherited from his mother. So when we look at IGF2 from the mother, it's going to be turned off. And if we look at H19 from the father, it's going to be turned off. Same thing in this female, right? So this is the copy that she got from her dad. This is the copy she got from her mom. Again, IGF2 is turned off in mom. H19 is turned off in dad. So when the germ cells are being produced, the methyl groups are modified so that they're removed, right? They're demethylated. And so that reactivates all of the genes. These are during production. And this is a male. This would be sperm. It's making sperm. So all these are turned back on, right? It's demethylated. So this is on. This is on. Then it's rewritten before the sperm is packaged and the gametes are made. The remember the dad turns off all of his H19, right? Because in your dad, you inherit a shut off copy of H19. So in the sperm, the H19, no matter what it is, what chromosome it's on, those are both shut off. They're remethylated. These IGF2s are both turned on. So when the sperm is made, H19 is off in males and IGF2 is on in males, right? This is the sperm gamete. Same thing happens with mom. So this is rewritten, right? The methylation is reversed. Then it's remethylated so that only IGF2 is methylated. And so when the egg is produced, only the IGF2 is turned off. H19 is turned on. This sperm fertilizes this egg. This is the parent. This is the child that receives those. Well, this is the zygote, right? This is the zygote, and then all of this is going to be passed down. So this is turned off from mom, right? This is turned off from dad. And again, this is IGF2 silenced in mom. And so this is maternal imprinted. And then, IGF2 is on in dad. H19 is off in dad. So this is this. And then this is this chromosome. So dad's H19 is off. This is H19. And IGF2 is off in mom. So that's what we say. That's what we mean when we say genomic imprinting. Okay, so this occurs in humans as well. Just in the essence of time, I'm not going to go really into this, but remember, these are the methylated islands, the CPG islands. This is just showing the pattern of methylation that occurs. I don't expect you to know this. I just want you to know that this is relevant. Again, this is how we do dosage compensation for the chromosomes. So remember, we have hypertranscription in Drosophila. So this is double expressed in males, right? Because they only have one copy of the X, their XY. In females, there's two copies. So this is the dosage compensation. In mammals, right, one of these is inactivated. So we get 1 unit of X product. This one is 2 units, 2 units. So they're all equal. And then we have hypotranscription. So in the female, it's reduced by half, where in the male, there's no Y chromosome, and so it's double expressed, so that they're all equal. Males and females have the same dosage of gene product from uh, their chromosomes. And then this occurs from uh, histone acetylase, uh, acetyltransferase. So this is going to add acetyl groups, right? And that activates chromatin. And then we can methylate it, and that would inactivate it. Okay, so we can see inactivated mammalian X chromosomes. They're highly condensed. We talked about their heterochromatin earlier. We can see them under the microscope, and these are called bar bodies. Um, the marks on inactivated X chromosomes, and then this is in mammals, uh, is the methylation of histones, the H3 subunit of histones, and hypermethylation of uh, its DNA itself. So remember, methylation inactivates chromosomes, so we would shut down the X chromosome. So in females, we have two X's, in males, we have an XY. One of these is going to be shut down in females, so that we each, male or female, get one dose of X uh, chromosome products. So this note, it just says X chromosome inactivation, as well as genomic imprinting, are monoallelic inheritance. So that means that no matter what is on this chromosome, right, it's off. So you're really only getting one active allele. And that's what we mean by monoallelic inheritance in diploid, right? Two copies, diploid organisms. Okay, so this is that, again, this is the, I talked about this. This is the, um, and I have to check this link, but this is the Neil deGrasse Tyson video about looking at epigenetics. It's a Nova video. I'm not going to show it because it echoes on my computer, but you can watch it. Okay, so we have post-transcriptional regulations of eukaryotic genes. Um, and that means, so we've made the transcript, right? We've already taken the DNA and turned it into RNA. So this is after it's been transcribed, and we can do alternative splicing, which we already talked about, and we can do RNA interference, which we've already talked about. And we've already shown this slide, so but we can alternatively splice. This is just showing this is myosin, the motor protein that powers your muscles. So this is just showing you what it looks like, the same gene looks like in different tissues: smooth muscle, striated muscle, brain, fibroblasts, which are kind of skin cells. All right, RNA interference. We talked about this already, the mechanism behind it, but um, the story behind this is this guy, Richard Jorgensen, who's at the U of A, University of Arizona. He was trying to make a blue rose. All right, so blue roses don't normally exist in nature. There's kind of like an X prize for this of like multi-million dollars if you can develop a blue rose. And so scientists have been able to do this. They inserted copies of gene to make things blue, but rose genetics is complicated, and so instead of just the petals being blue, petals, the whole thing is blue. So the petals weren't just blue. The whole, so we're talking about leaves, thorns, everything is blue, and that's not really the goal, right? So what he was trying to do is insert a blue gene into petunias that have less complex genetics to prove to investors to give him more money to do research so he could make a blue rose. So to show proof of concept, he added purple genes to make petunias more purple. Um, and what happened is when he added this purple gene to make it more purple, instead they came out white, which is kind of the opposite of purple, right? And this is similar to Mendel's thing. So the default pathway is white. If you have a gene that makes a pigment, then that pigment is positive, so the flowers are purple. And so anyway, this is how RNA interference was discovered, and we call it RNAi. And basically, what he did was he flipped the gene in backwards, and we didn't really have a chance to cover this, but we did talk about it in lambda-phage. And in lambda-phage, it can have so this DNA can be 5 to 3, and this would be 3 to 5. So DNA is always read on the 3 to 5 strand in this direction, right? So we can make an RNA transcript from the same region of DNA on both strands. You just need a promoter here, promoter here. And I kind of showed you guys this in the beginning. So he flipped the gene around. So what he did was he made an RNA, which was a reverse complement to the normal RNA. And when he did that, the two RNAs bound together. And this double-stranded RNA to a eukaryote looks like a virus, right? Because we normally don't see double-stranded RNA unless it's in a virus. And I kind of showed you guys that earlier too. This activates RNA interference, which destroys all of the RNA, no matter if it's good RNA or bad RNA or a different RNA, and it destroys it all. So the RNA that codes for purple is destroyed, and so no purple is made. And if no purple is made, then the flower is white by default. And so the idea is that you can do this. You could put in double-stranded RNA for almost anything and shut that translation, post-transcription, shut it down. Silence those genes. So it's an experimental method to turn off genes by injecting double-stranded RNA, into plants or animals. And sometimes it works, sometimes it doesn't. There's actually a video on Nova, if you look it up, RNA interference, and I think I put the link in Canvas under Pages. So if you want to explore this more, they use this for macular degeneration. So in macular degeneration, you have blood vessels that there are too many blood vessels in your eyes and they leak, and so it kind of looks like looking through a cloudy window, or cloudy, sunglasses or smudged sunglasses. So the idea is that if you put in the gene to make more blood vessels and you make it look suspiciously like a virus, then it'll trick the cell into turning off blood vessel production, and that'll clear up your vision. So they've done this. They've injected directly RNA, double-stranded RNA, into people's eyes that have macular degeneration, and it does appear to be helping. So it's a promising technique. I think we covered this, but I'll do it again. So whenever a cell is introduced to double-stranded RNA, it thinks it's a virus, and because that's weird, and it activates a protein called dicer. Dicer is an endonuclease. So an endonuclease is something that cuts on the inside, so it's going to cut this. And a nuclease is DNA, cleaves DNA or RNA, nucleotides. So dicer cuts up the double-stranded RNA into what we call small interfering RNA. So little pieces of RNA. These little pieces are bound to what we call the RISC, which is the RNA induced silencing complex. Um, and so one of these strands is tossed, the other strand is retained in the RISC. And so what RISC does is then it goes through the cell looking for target mRNAs. When it finds them, it binds to it, and it has an endonuclease in itself. And when it does that, it will start chopping up the RNAs. Any RNA that matches this, it chops it up. And so, in our example, if this is the RNA for purple pigment, and it chopped it up, then we would get no purple pigment. So it would go to default, which in this petunia's case is white. Okay, so there's other things called microRNAs. MicroRNAs, they produce siRNAs that silence gene expression. Uh, they're pro- they're produced by regions of DNA. So there are areas of DNA in between genes that we used to think were junk DNA, and now we know that those are actually regions that can produce these microRNAs to silence genes. Um, they're not translated into proteins. Their job is to activate the RNA interference pathway. They make hairpin structures. So they code like this, and then this part is the double-stranded RNA. But these are quite lengthy, and this activates the RNA silencing mechanisms to shut down the targeted mRNAs. So that's another way that you can do gene expression. And this is just showing you this. So let's say here's our protein coding gene. It's going to be transcribed in this direction. This is our microRNA gene. It's going to be transcribed in this direction. It produces these microRNAs, which is shown here. So, maybe there's two stem loops, or maybe there's one. It doesn't really matter. And that's going to activate dicer. All right, so it can go. This can be outside and in to the cytoplasm. So this is the nucleus. This is the cytoplasm. So these are exported out of the nucleus as microRNAs. Dicer binds to that. It does the same thing with the RISC complex, and then that can silence the by chopping up the RNAs. The ribosomes can't actually transcribe the genes. So we get mRNA degradation and we get translational repression. Same time, because there are no more RNAs. And then there's an animation on this for YouTube of how these work. Um, and that's it. So that's the end of eukaryotic gene regulation. And then the last lectures next week will be, there'll probably be two on this. I might be able to fit it in one, but I will make lectures for this next week. Next week is the last week, and then you guys will have your final exam. So the goal is Tuesday and then Thursday. Make sure you guys go over the reviews, right? There's PowerPoint reviews for the final exam, just like there were for the second exam. So study that stuff. I made a couple quizzes for um, the subject matter that we've already covered. That's in Canvas, so make sure you take those quizzes. You can take them 3 times. Um, and that should be all the material you need to take the quiz that's on um, the tryptophan operon and the eukaryotic gene regulation. All right. If you have any questions, email me. Come to office hours.